Warm welcome to one and all. Now we are going to discuss with very advanced and one of the very interesting topic, bioinformatics. First, what is bioinformatics? What is the major definitions related to bioinformatics? It's a mathematical, statistical and computing methods that aim to solve biological problems using DNA and amino acid sequences and related information. That is bioinformatics. That means computed softwares to solve the biological problems. That is the mathematical and computational methods to solve biological problems. That is known as bioinformatics. Then major bioinformatics areas, which comes under computational languages, that is C, C++, Perl, BioJava and all. It's related to the genomics, and biostatistics, that's connected to the proteomics. Proteomics, that is the bioinformatics study related to proteins. And computational biology, database management system, and chemi-informatics and systematic biology. These are the major bioinformatics areas. And in silico areas of bioinformatics. In silico, that is the computational methods of bioinformatics, that's having different branches, that is comparative homology modeling, phylogenetic analysis, protein structures prediction, that is the protein structure related studies, and protein folding problems, and microarray analysis, assay methods, and docking approaches, and new drug discovery. These are the major in silico areas of bioinformatics. And major biological databases. We can move to the major biological databases. The biological databases related to the bioinformatics, that's having different types of databases are here. So, first one, bibliographic databases. In the case of bibliographic databases, it's mainly included the literature studies. And taxonomic databases, that's mainly for the classification related studies. And nucleic acid databases is related to DNA information. And genomic databases, that is the gene level, genetic information. And protein databases, that's the protein information, that is amino acid sequences. In the case of bioinformatics, all the informations are expressed in the manners of sequence manner. In the case of protein, that is the amino acids, there are different types of 20 amino acids. And in the case of gene, that is the DNA, different types of nitrogenous bases. And protein family studies, domains, functional sites and all, it's mainly connected with the classification of protein and classification of proteins and identifying domains and all. Then it's also included enzymatic studies, metabolic pathway studies and all. It's mainly connected to metabolic reaction studies. These are the major biological databases. And types of biological databases. Based on the functional studies, based on the structural studies, the biological databases are divided into three important ways: primary databases, secondary databases, and composite databases. In the case of primary databases, that's mainly sequences in the form of nucleic acids and proteins. It's having EMBL, European Molecular Biology Laboratory, and GenBank, DDBJ, DNA Data Bank of Japan, SwissProt, TREMBL, PIR, Protein Information Resources. These are the major types of biological databases. And next to move to the secondary biological databases. In the case of secondary biological databases, having ProCytes, blogs, prints, PFARM and all. PFARM, that is protein family studies. And third category, composite databases, it includes NRDB and all. These are the different types of composite databases. The major primary, secondary and tertiary databases, that means DDBJ, EMBL, GenBank, it's a interconnector works, it's a collaborator works. That's why providing the sequence information to publics and research communities. In the case of these intercollated databases, the main category, that is the NCBI, National Center for Biotechnology Information. So National Center for Biotechnology Information, the main website link, that is the www.ncbi.nlm.nig.gov. This is the main link address, is connected to the NCBI. So NCBI is established as the government's response to the needs for more and better information processing methods to deal with a vast amount of data. It's mainly developed by NLM, that is the National Library of Medicine, and NIH, that is the National Institute of Health. It's a comprehensive website. It's including biology related databases information and tools for viewing and analyzing and automated system for storing and retrieval of data. In the case of NCBI, along with the EBI, European Bioinformatics Institutes, and CIB, Center of Information of Biology, together to form international sequence databases collaboration. So it's mainly having GenBank, EMBL, DNA database of Japan, that is the DDBJ. These are the main homepage of NCBI. It's having different searching options are here. All databases and different resources, popular and featured, many types of information available in this specific NCBI web page. This is the overview related to NCBI. It's having all databases, different types of informations, PubMed, protein and nucleotide related information, GSS information, EST and bio projects, different types of information available through the particular NCBI site. And this is the entrance homepage related to the NCBI. It's have a lot of information here mentioned. PubMed, that is the literature level studies and all databases, human genome, GenBank and MapViewer and all. So when we are searching a single with the accession number, otherwise single organism, here plan mention Oryza sativa. When typing the Oryza sativa, entering the Oryza sativa and so provide the search button, we will get the all the types of information related to Oryza sativa. And another important, it's like a GenBank, EMBL, it's the providing most important searching tool, database, bioinformatic tool, that is the BLAST, that is Basic Local Alignment Search Tool. It's a predominant sequence alignment tool for performing rapid searches of nucleotide and protein. Sequence databases, nucleotide and protein database search is mainly used for homology searching. Homology, that means similarity sequence searching tool. And it's a mainly detecting local as well as global sequence alignments between the query and database sequences. Then we can move to some few exercises related in bioinformatics. The first one, to search nucleotide sequence data of Oryza sativa, that's a rice, and download the data into separate text file in the form of GenBank and FASTA format. FASTA, that is a fast all. This is a main page, NCBI. We are typing here Oryza sativa and provide the search options. So that time we will get a lot of information related to Oryza sativa. It's having lots of information, so protein related information, genome related information, taxonomic information and SNP, single nucleotide polymorphism studies and all. We will get lot of information related to the particular Oryza sativa. Then after that, this is a main button. So here different option is available, the GenBank, FASTA, graphics and all, and we can click the GenBank format and after that we can select the FASTA option. Then this is the main NCBI, National Library of Medicine homepage. So we are providing the Oryza sativa and search options. So this is the BLAST tool. So we are collecting the sequences and paste here the FASTA option, the BLAST options. We will get the similarity searching tool, the specific sequence related, all the information is available through this way. This is the searcher BLAST related information. So here mentioned Oryza sativa, the sequence similarity, 100% sequence similarity, all the types of information is available through this BLAST options. That's all about the first exercise. Then we can move to the exercise 2. So here, to view the genome map of Oryza sativa with the chromosome number. How can we visualize the chromosome number of the specific Oryza sativa? Okay. Then This is a entrance page, NCBI. Then we are providing the name Orisa Sativa here and click the map view button. And click map view button. It's having the lots of information related to Orisa Sativa. This is the graphical chromosomal representations related to Orisa Sativa. Then another tool is after BLAST is another one, another important bioinformatic database, that is the EMBL, that is the European Molecular Biology Laboratory. This is located on Wellcome Trust Genome campus in Kingston, UK, along with the Wellcome Trust Anchor Institute. Then career prospects in bioinformatics. What are the major career options in bioinformatics area? So it's providing scientist, research associate, professor, lecturer in universities and colleges, technical executives in business and development, data analyst in science and organizational laboratory, then biosoft developer in the IT industry. Then major applications of bioinformatics in different fields. What are the major areas utilizing the bioinformatics works? We can move to that. The first one, molecular medicine. Most widely used in the field of molecular medicine. This is a different manner, more direct targets, personalized medicines, preventative medicines and gene therapy. The first one, direct targets. Use these bioinformatic tools. So it's an improved understanding of disease. Specifically, we can identify the specific gene disease level, molecular level of the disease, and this provides protein level and the compound drug interactions, gene expression profiles, and phenotypic study and clinical records. So all the data we can perform through the way of bioinformatics. It's a very interesting and very advanced area in drug targets. And after personalized medicine, today's doctors have to use trial and error methods when prescribing the medicines. When we identify the sequencing studies, we can easily provide the exact or specific medicine in the specific diseases. This is one of the most important area in bioinformatics. In the case of preventative medicine, so one of the cases that is a vaccination process. So most advanced that is the edible vaccine. The vaccination providing in the edible fruit. Raw edible fruit is like banana and tomato, carrot also providing the vaccine. So easily we can use. It's a preventative medicine method. It's only possible through the bioinformatics sequence studies. Then gene therapy. It's another important area, another applications related. Gene therapy is approach used to treat or cure even prevent disease by changing the expression of person's gene. We are changing the gene expression and curing the disease. This method that is known as gene therapy. That's a therapeutic transgene, then providing into the target gene and produce therapeutic protein level. And another important area, that's the microbial genome applications. In the case of microbiology field, we are utilizing different applications here. Waste cleanup, then climate change studies, alternative energy sources, and in the case of biotechnology also, and antibiotic resistance, forensic study, and bioweapon creation, evolutionary studies, and all. These are the major areas related to the microbial genome applications in bioinformatics. This is a waste cleanup method. So we are providing a very potential microorganism. We are developing potential microorganism to treat the waste processing, waste cleanup processing, the different industrial level waste and agrochemical waste like excessive use of fertilizers and all, and waste disposals like a sewage and sludge and all, then mining activities, all the harmful polluted created components we are degrading with the help of the potential microorganisms. Then climate change studies also we are utilizing the bioinformatic areas. So this way to understand the climatic factors. Then forensic analysis. It's providing the Bacillus anthrax is one of the common organisms. Scientists used the genomic tool to help the distinguishing between the different varieties of strains. And bioweapon creations. Most advanced area, bioweapon creations. So another important applications in bioinformatics. Then it's also utilizing in the evolutionary studies also. Evolutionary, the molecular level studies also we are utilizing. Then another important area, that is the agriculture. In the manner of agriculture here, we are using crop productions and improved nutritional quality, then growing poorer soils and drought resistant plants. So here mentioned some example here. So when single plant that's having potato and tomato, that is known as tomato, and seedless grapes, and seedless watermelon, seedless pomegranate, seedless lemon and mango, and a flavor saver tomato, that is the delight food ripening tomato. These all the components we are developed based on the sequence bioinformatics sequence studies. Some most important applications in the area of bioinformatics. We are utilizing the agriculture field also. Then in the case of agriculture here, insect resistant plant. That is insect resistant, the most common, that is the Bt cotton, Bacillus thuringiensis cotton, Bt brinjal, and all we are developed. Most insect resistant plant. Then improved in nutritional quality. So in the way of improved in nutritional quality, we have developed the rice, that is the golden rice. It's having vitamin rich rice. The rice that's having rich of vitamin. That is the most improved nutritional quality product in the way of agriculture through the method of bioinformatics. We are utilizing the method of bioinformatics. The rice that's having rich in vitamin A. In the manner of animals, we are providing yolkless egg, that is only protein egg, and lactose free milk, fat free milk, and all. Then therapeutic milk, therapeutic protein, therapeutic meat, and all we are developing. Then comparative studies. In the case of comparative studies, we can study the evolutionary changes. Different organism evolutionary studies also analyzed in the method of sequencing or phylogenetic analysis tree. This is the method here mentioning the phylogenesis. It's not only the physical appearance, it's a molecular level studies based. We can easily to construct phylogenetic tree. That's all the basic things related to the bioinformatics, and we can move to some important questions in bioinformatics. So name the scientist created the first bioinformatic database. It's the Margaret Belair. So she mentioned as father and mother of bioinformatics. It's March 11, 1925. It's an American physical chemist, primarily study related in bioinformatics. Okay, next. The identification of drug through the genomic study is called, that is the pharmacogenomics. It looks at how your DNA affects the way to respond to drug. Then moving to the next question. The term bioinformatics was coined by Pauline Hoogweg. It's a Dutch theoretical biologist that studied the complex system related bioinformatics. So the particular scientist Pauline Hoogweg provide that name bioinformatics. Bioinformatics, that's the combination of biology and computer science. The next question. The laboratory work using computers and associated with web-based analysis, generally online, is referred to as the in silico method. That is a computational method. And okay, the next one. The process of finding the relative location of gene on a chromosome. How to identify the relative location of the chromosome. Gene in a chromosome, that's known as genome mapping. Through the genomic mapping method, we can identify, we can point out a single gene, a specific gene present in that entire chromosome. The next question, human genome contains approximately dash base pairs. Human genome that's contain 3 billion base pairs. So one more, 3 billion base pairs. This is the entire chromosomal structure, and it's having total 3 billion base pair. Base pair, that means nitrogenous bases present in DNA, that is adenine, cytosine, guanine, thymine, and all the 4 types of nitrogenous bases. So the total that's having 3 billion base pairs. Okay, the next. Give an example of homology and similarity tool. So homology and similarity search tool, that is the BLAST, Basic Local Alignment Search Tool. There are different types of BLAST is available. Nucleotide BLAST, protein BLAST. In the case of protein BLAST, we can search the protein related similarity sequences, similarity organisms and all. In the case of nucleotide related, we can search the nucleotide BLAST. Then different types of other BLAST is also there. BLAST X and T BLAST and all. That is a translated DNA to RNA, RNA to protein, the translated blast works. And what is the deposition of cDNA into the inert structure? That's called DNA microarray methods. In the case of DNA microarray methods, we can identify the entire sequence studies, cDNA studies. Okay. Then that's all the basic things in bioinformatics. These are the major conclusions. We have discussed about the basic introduction to bioinformatics and major definition related to bioinformatics and major areas of bioinformatics and biological databases like NZBI, EMBL and all. Then career prospects in bioinformatics, then major applications of bioinformatics in different fields.